Okay, go ahead.
Yeah, thanks, Steve, for the introduction. And I'm glad to share my work with all of you working on the similar area. So today I will introduce some work on genomics, genome sequencing, benchmark suit, and accelerator design. So today I will basically introduce three parts. First, some backgrounds for DNA sequencing if you are new into this area, and then the genomics benchmark suite, and finally an accelerator design. So if you are interested in this area, feel free to um, email me for further talk. Here is my email. So the high-level background is the precision health in this field. Precision health is an emerging approach for disease treatment and prevention. It means that there could be a medication suitable for everyone. However, if the medication is targeted to different people based on their different genes, there could be potentially better treatment performance. Then in this process, the human genome plays an important role in the personalized treatment. In the past 2 decades, we could see uh, the genetic data is produced cheaper and faster. The cost to obtain per human genome has been decreasing from millions of dollars 20 years ago. That was the first time the human genome could be sequenced. And up to like a couple of years ago, it has been dropped to hundreds of dollars actually. Also, if we look at the sequencing database, we could see it has been increasing exponentially in the last 10 years. Therefore, this explosive growth of genetic data motivates the hardware acceleration for such genome sequencing analysis. And that's our target today. So first, let's look at some backgrounds. So DNA is a polymer composed of two strands. Each nucleotide base is represented by ACGT characters. DNA sequencing is a process of identifying each of the base pairs within these DNA strands. Here are some commonly used genome sequencing pipelines. The first one is reference-guided assembly. The reason people use a reference to acquire each person's genome is that the difference between the genome of two people is very small. It's usually less than 0.1%. So that's why we could first build a reference for the entire human being and then test the sample DNA for each individual against this reference. And this will reduce the difficulty to sequence the entire DNA sequence for each individual lot. So it makes it much easier to sequence from scratch. The first step is to acquire the DNA sample from some tissues like uh, here, like the blood or some other tissues. Then from the sequencing machine, we could acquire the some electronic signals. Then we use some base calling algorithm. Today, most of them are machine learning based to convert these electronic signals to ACGT base pairs. And these ACGT base pairs are usually composed in reads. Reads means from hundreds to 10,000s of consecutive small strands because the long chromosome is usually broken in this sequencing machine. So after base calling, we acquire some reads with length of either hundreds of base pairs or 10,000 of. The next step is to map these small reads to the long reference. The reference length is usually like 6 gigabases. So after we map each of the reads to the reference, then we detect a difference between each individual sample and the reference. And we name this comparison the variant calling. So if there are some difference, then we name it a variant. So in the end, we will acquire a variant calling file. And this file records the difference between each individual's gene and the human genome template or reference. And that is actually the each individual's genes that we want to we we want to know. Then there is a question that what if we don't have such a reference? And that is a big problem. For whatever new species, we have to build such a reference, and that is a much more difficult problem than the reference guided assembly, because we will get like thousands or millions of such reads, but we have no idea what the order we should to assemble such small reads. So in this case, what people do is to find the neighboring reads. Neighboring reads means if two reads each has 100 base pairs, then we find two reads. Between them, 99 base pairs are exactly the same, and only one is different. So then we know, okay, these two reads should locate nearing with each other. So in this way, by looking for some reads locating near each other on the reference, we could build a huge graph. So in this graph, each node represents a read, and each edge points to a large overlap region. So in this, we build such a large graph very, I mean, it is a huge computational problem. So that's why people don't want to build such a graph every time to sequence a DNA sample. Then we have some algorithm to find a consensus from this graph to linearize this graph to a single sequence of reference genome. In the end, we will do a lot of error correction and polishing because this reference will be utilized for the entire species. So we have to reduce the error because in this sequencing process, there could be some errors introduced easily by insertion, deletion, or substitution of base pairs. There are also some other applications to classify different species among a sample. For example, when we get a sample from human, it does not only contain human DNA, it could also contain some virus, bacteria, and so on. So sometimes people want to estimate the abundance of different species among a sample. So these are some commonly used sequencing pipelines. And among them, today, one of the most common application or clinical application is the first reference guided assembly. So in this reference guided assembly, an important step is this read alignment. Read alignment means we want to map each read to a specific location in the reference genome. So let's first briefly look at how read alignment works. Here is a small read and a large reference. We usually don't want to map the entire reads like from hundreds to 10,000 of base pairs because there are a lot of insertion, deletion, and substitution. So it's pretty hard to do an exact string matching for a long string. So what people usually do is to find some small seeds. Seeds means an even shorter string. Then for seeds, we could easily do such exact string matching without any error. For these seeds, we could find multiple candidate position. And from the this mapping of seeds, we extend to both direction. When we extend them, we could tolerate some error. Then we name it approximate string matching. When we extend the seed from both direction, we will evaluate the quality of this extension and give them some scores. For example, in these three different locations, when we look both left and right direction and compare the reference and the reads, we use this score to evaluate how different they are. The less difference, then the score is higher. For example, the third position actually has exactly the same characters between the read and reference, so the score is the highest. And this process is seed extension. So read alignment is actually achieved by seeding and seed extension. So in seed extension, as I said, the approximate string matching is used to evaluate the similarity between the read and reference. Like here, there are deletion, insertion, and substitution. So the added distance or the similarity could be defined as 4. The way to calculate this similarity is to put read and reference to two sides of this scoring matrix. And here we use a dynamic programming algorithm named Smith-Waterman to calculate the score. When we compare the one pair of character, one from read, one from reference, if they are the same, we give it a reward of a positive value. If there are difference, it means there are substitution, insertion, or deletion, then we give it a penalty as a negative value. And when we fill out this table in a dynamic programming way, we look for the highest score and trace back to the start point. In this way, we could find we could match the read and reference and find the difference between them. And this compute complexity is O(n) square. There are also some optimization, like banded version. Banded version means if we find this path, it usually locates along the diagonal. So people could ignore the upper and bottom triangle to reduce the compute complexity to OWN. W is this bandwidth. So here are some backgrounds of DNA sequencing. And then we could look at a benchmark soup design. But before that, feel free to ask any question.
So when you're doing the matching,
Mm-hmm.
you end up like committing to one of them kinda, is there? Cause if there's, say, like 2 differences in one and 3 in the other, and they're close, and you end up going for the lower one, is there an error checking later for you happen to have made the wrong decision? Or is that kind of, you're committed to it at that point? Like the previous slides where you were doing the multiple, yeah. So say 103 is the correct site, but you pick 512 or something. Is there a way of knowing for sure that you're right? Or is that score? You know what I mean?
Do you mean it is possible that we cannot find the exact match? So all of the candidate positions have some errors, right?
Yeah,
Yeah, that's very possible. So because the chromosome is very, very long,
yeah.
and there could be like more than 100 possible position. It's possible that none of these positions get perfect match. It's also possible more than one location has perfect match. So
Mhm.
both are possible. So after we map each of the reads, there are also some subsequent algorithm to assemble all of these reads together. For example, uh, we not only have this read. For example, this read will be mapped to here, to this location. Usually we will have another read here, cover most of these reads and some other on the right part. So, and there are also maybe this read. So when we considering these three reads together, then we will have like three map. Uh,
Oh, gotcha, okay,
yeah, then we could have a more comprehensive, I mean, matching result.
okay, that makes sense.
Yeah, that's a good question.
So I have a follow-up question. So is this part, this kind of overlapped realignment, something you can call it consensus based?
Uh, so what what is
So
consent?
previously you mentioned that consensus. So I'm wondering
Oh, okay.
whether this part is also
Oh, I
one
see.
some sort of consensus.
I see, I see. Yeah, yeah. So the last consensus I think I okay, sorry, let me clear that. The last consensus is used in de novo assembly like to build the reference. So here consensus is I mean a necessary step. But here it's possible that we couldn't find like the exact or like say perfect match. But here the concept is not, I mean, necessary. Uh, because that will introduce a lot of computational difficulty. But here, without such consensus uh, process, we could already get like maybe like 99% accurate result because we have this reference. So this reference used to reduce the workload. So it's actually true if we do more work, we could get another like 0.5 percentage of accuracy. But in clinical, maybe people don't want to do that. It's a trade-off. We do more work, it will be accurate.
Okay, makes sense.
Yeah. So in order to explore the hardware acceleration opportunity, people usually build benchmark. So in this benchmark GenomicsBench, we collect some time-consuming algorithms from these commonly used sequencing pipelines. So here we could see there are multiple algorithms extracted from different applications like read alignment, variant calling, and so on. So the reason we extract this algorithm is that they takes most of the runtime among these software pipelines. And we collect their algorithms, like some are dynamic programming algorithms, some are matrix multiplications. And then we analyze their performance characterization on commodity hardware. So the first analyze we did is the instruction diversity. We use the Intel Pin 2 to collect the types of instructions. Like here, some are optimized by Intel AVX, SIMD instructions, so we could see a lot of vector instructions. And we also use the Intel V2 profiler to export their microarchitectural patterns. So the second figure shows the pipeline stall caused by different reasons. Like some kernels are backend core bound. It means the pipeline stall mostly are caused by the lack of compute logics. It means these algorithms are compute bound. But some other kernels are memory bound. It means they're mostly bounded by memory access. There are also some kernel are bounded by front end because there are a lot of branches if we look at the source code. So there are some bad speculation and wrong branch prediction and fallback. We also analyze the memory pattern like cache misses and the percentage of CPU cycles waiting for data, and how many DRAM bits it requires for kilo instructions. Among this analysis, basically, there are some information we could acquire. So first, are these algorithms either compute bound or memory bound? And that will give us a high-level idea what architecture we should choose to satisfy this workload. And also this benchmark suit will create a somehow standard test bed like for accelerator design. After building a benchmark suit, then the next or the most important task we want to do is to design an accelerator or some accelerator solution on commodity hardware. So here are the 3 steps for the reference guided assembly. And there are multiple software tools if we want to finish this process. On general purpose hardware, there are some comprehensive libraries to accelerate all of these different softwares. Like Intel provides a library to utilize their SIMD instructions to accelerate all of these different steps. And NVIDIA also has their toolkits to best utilize their GPU hardware to accelerate these algorithms. Also, there are a lot of custom accelerator design. They show much better performance than the general purpose hardware, but they also have much higher design costs. It means whenever there is a different algorithm coming up, then people have to design a new accelerator because most accelerators only target one algorithm. If we want to utilize the accelerators to perform the entire software pipelines, then we have to think of how to connect these accelerators. So there are questions or people will think of, is there any solution sitting between these custom accelerators and the general purpose hardware? One solution is the domain-specific accelerator. If we could program this accelerator and make it less custom, or we could sacrifice some performance, it may be able to bridge the gap between the general purpose hardware and the custom accelerator. So the next work I want to introduce is GenDP. It's a dynamic programming accelerator. When we build the benchmark suit, we observe that there are multiple algorithms using dynamic programming algorithms, and they are all time consuming in these software pipelines. So we want to build such a programmable accelerator to solve dynamic programming, because that is a fundamental algorithms used in many places in this genome sequencing software pipelines. Before introducing this work, I want to show that this dynamic programming is actually an important algorithm. For example, in NVIDIA's Hopper architecture, they introduced DPX instruction. It's a specialized IC to it's a kind of, domain specific optimization on the GPU. So in this IC extension, what I acquired from the CUDA API is that, so there are max reduction, max followed by addition or comparison. So these instructions are commonly used in the genome sequencing algorithm. So these operations could be integrated to some special IC, and on H100, this DPX instruction could introduce some extra performance gain. So let's first look at what is dynamic programming algorithm. In this example, we want to calculate the score of the entire table. And we know the initialization, that is the scores in the first row and first column. It's similar to the Smith-Waterman algorithm that I mentioned before. And we know the objective function that is based on some known result, how to calculate the new score. So based on the objective function, we could finish the entire problem. And this large problem is actually broken down to some simpler, smaller problem. So then let's jump into some genomics dynamic programming algorithms. And the first one is Bandit Swiss Waterman that I mentioned before. So these two algorithms are pretty similar to the previous small example. So when we calculate a new score, this score depends on some results on the left, upper, and diagonal neighbors. And there are also some algorithm related to the graph. So in the third algorithm, we could see we want to map a sequence to a graph. So this graph is linearized in this table. For example, we could see the node AACGCAGC. And we map all of these nodes sequentially in the first column. So when we consider mapping a sequence to a graph, this dependency pattern is a little different from the previous two, because there are not only the dependency on left, upper, and diagonal neighbors, there are also some dependency far away. They are dependent on the graph structure. Say, if there are an edge connecting two nodes, then we need to consider this dependency. For example, for this G is connected to both C and A, this C and A. So that's why when we calculate this new score, we have to compare with the previous row and the next previous row, because there are two nodes connected to this G. Also, there are some one-dimension dependence table. So when we calculate a new score, we need to consider the several previous neighboring scores. If we summarize these dynamic programming algorithms, we could see that they're very different. So they have different problem size. So these examples I show are pretty small tables, either 1 dimension or 2 dimension. But in reality, for example, this 1 dimension table could up to 20,000. And this 2 dimension table, some are small, some are large. Their dependency pattern are different. Like some are very simple as the first two kernels. Some are complicated because they are graph structure. Also, their objective functions, the operations we need to calculate are very different, and the precisions are different. So this divergence of dynamic programming algorithms make it very challenging to design a domain-specific accelerator to support all of these different characteristics. And our solution is to build custom hardware for their similarity and use programmabilities to support their difference. So GenDP includes DPAX accelerator and the DPMap algorithm. When we have a new dynamic programming algorithm, we first analyze this problem and look at its intercell dependency pattern. And then we analyze the compute patterns, that is what operations it requires, and use DPMap algorithm to convert the data flow graph into compute instructions. And I will introduce more details later. So let's first look at DPAX accelerator. It has the systolic array as the base architecture. There are 16 integer P arrays and 1 floating point ones to support the different precision requirements. The integer P arrays could be concatenated to form a larger array to support the different dependency pattern of these algorithms. When we map a 2-dimensional DP table to DPAX accelerator, we map each row to each PE. So each P array is composed of 4 P's connecting one after the other. And there is a FIFO connecting the first and last P. In the left table, each row in this DP table starts at different time. So we need control instruction to tell each P when to start computation. And the control instruction also controls the data movement between different P and between the P and the input-output data buffer. So the control instruction also, okay. Yeah, so that's the P array. And then let's look at each of the PE. On the left table, the cells in each row is mapped to a single processing unit. The objective functions is converted to the compute instruction. The compute instruction is executed on the compute unit array. There are 8-bit and 32-bit compute unit. So we designed a SIMD architecture to support the different precision requirements of different algorithms. There are also control instruction. They controls the data movement between different components, like the register file, the scratchpad memory, and the data movement between this component and the input-output ports. The control instruction also pipeline the data movement and the computations. So this is the processing unit architecture. Then as a design choice takeaway, let's see how this similarity and difference among these dynamic programming algorithms is supported by the architecture design. So the first similarity is the local dependency. Most of the dynamic programming has such local dependency. Local dependency means the computation for one cell usually depends on some previous results, and these previous results are not generated far away from the new problem. So we use the one-dimensional systolic P array with a FIFO as a base architecture. And then from the objective function, we observe that there are lots of reduction tree data paths. So if we recall the NVIDIA's DPX instruction, so they also support a similar IC based on this reduction tree data paths. So we designed the compute unit similar to this data path. Then let's see the difference between these DP algorithms. So first is precision. We design both integer and floating point P array. And in the integer P array, we have the SIMD compute units to support either 8 or 32-bit compute units. And if we combine the two compute units of 8 bits, it also supports 16 bits. The second difference is the dependency pattern. So we combine the 16 integer P arrays to concatenate one after the other to form a larger array in order to support both 2-dimension and 1-dimension the DP table. And the third difference is the long dependency. So if we recall the graph structure, if there is an edge connecting to nodes far away from each other, then here the local dependency is not enough to support such pattern. So we introduce a software managed scratchpad memory to temporarily store such intermediate value to support the long dependency. And finally, the different dynamic programming algorithms have different objective function data paths. So we introduce an IC rather than very custom operators and data path. So that's how we use the similarity to customize the hardware and use programmability to support the difference between these algorithms. And then let's look at this DPMap algorithm. So basically, when we have a objective function, we could convert it into a datable graph. And we break this datable graph to map it on the compute unit. And that's how we map different dynamic programming algorithms to our compute units. So there are 3 steps. It's kind of complicated, so please refer to the paper if you are interested in the details. We evaluate several dynamic programming algorithms on this GenDP accelerator and compare it with the CPUs and GPU baselines. So for GenDP design, we obtain the throughput from a cycle-accurate simulator and do synthesis to estimate the power and area. We use the throughput per unit area as a matrix when comparing to GPUs and other accelerators. So it achieves like more than 100 times throughput per unit area when compared to the general purpose hardware. However, if we compare it with some custom accelerators, it's actually not as fast as custom accelerators. The reason is this programmability actually sacrifices the performance. So it's impossible to support multiple algorithms and faster than some custom accelerator supporting only a single algorithm. So it's a trade-off between the customality and generality. And we also demonstrate that this GenDP supports dynamic programming algorithms in other domains like speech detection and robot motion planning. Finally, I want to discuss the generality and limitation when we compare different types of hardware. If we look at general purpose hardware, it supports everything like GPU. And the normalized performance, we name it 1. Then GenDP or soft brain are domain-specific accelerators. So they're programmable. So they support lots of different algorithms. But in some algorithm, there are limitations. The limitations means it could not efficiently support them. And the performance of these domain-specific algorithms are usually much faster than the general purpose ones. But if we look at the custom accelerator, they usually support only a single, and they're usually much, much faster than the general purpose or domain-specific algorithms. So this table shows a trade-off between the generality and customality when we want to design an accelerator. So recently we are also working on some extension for GenomicsBench, like looking for some new algorithms. Previously, people usually map some DNA samples to reference sequence. And recently there are some reference graph because in human being, there are different like there are white people, black people, and each subspecies could have their own reference. So it's not enough sometimes if we only have a single reference to represent the entire human being's genome. So when we have multiple reference, people could merge them into a graph, and we name it pangenome graph. So this pangenome graph could better represent the the commonality of the entire human being rather than using a single reference, because on some position like white people or black people could have different variants, and this graph could better capture these patterns and reduce the bias. And our previous hardware analysis focus on Intel-based hardware. And there are also some ARM-based profile. So this is also an exciting ongoing project for the GenomicsBench extension. So that's mostly about my my talk today, and feel free to ask any question or email me if you want to further talk about that.
Okay, cool. Any questions for online? So maybe let me start with the first question. I saw that, for example, when you're doing the dynamic programming, you are supporting both integers or floating point. So my question is why the floating point is needed for dynamic programming? I saw the algorithm seems working on mostly the addition, the maximum, those kind of base operations. So why the floating point is needed?
Oh, yeah, that's a good observation. That's correct. So most of the algorithms are integer based. That's why we have most of the resource allocated for the integer operation. The floating point, if we look at this summarization table, we could see this pairwise hidden Markov model utilizing variant calling is using floating point. So usually when people use floating point, it means we need this precision. We don't want to lose precision. So this pairwise hidden Markov model is utilized. Let's go to the, okay. So this algorithm is utilized for error correction and variant calling. So this problem is not a very deterministic problem, like there is a single answer, but it's a probabilistic problem. So for variant calling, the result is usually okay with high probability. This position, what is the in this position, what is the variant? Like either it's the A changes to C or T. Because there is no deterministic answer, so people want to get a result as precise as possible. So in this algorithm, it requires the floating point operations. It, yeah.
Okay.
Yeah, so here it's most like a probability calculation rather than a string matching problem.
Okay, that makes sense. So that floating point, the IP32, right?
Yes.
Okay. Okay, any other questions from online or people here?
Hey, this is Dr. Wang. I listened to this talk late, but I'm curious, because this genome sequence analysis has been a while, for a long time, right? Without we talk about these accelerator computing, domain specific accelerators, and I know MapReduce and the Hadoop. When we talk about big data, like 10 years ago, 15 years ago, right? People already use these Hadoop, right? MapReduce to accelerate these genome search, and people have this MPI program and MapReduce platform develop these parallel program to significantly improve this genome sequence analysis search. So how do you compare this hardware accelerated approach as compared to this large scale distributed data processing platform like Hadoop?
Yeah, that's a good question. So as you said, the MPI or this uh like distribution among different nodes are commonly used in the database or distributed system. So in genomics, this problem size is not as large as the workload you described. For example, there are some problem. The size is large enough, we have to distribute it on multiple compute nodes. Meanwhile, we need to we need a good communication protocol between these compute nodes because all of the intermediate value are correlated with each other. So in genomics algorithms, most of the parallelism are independent. For example, if we want to like in like here, if we want to map a read to reference, Usually there are millions or billions of reads to map to this reference, and all of these mapping are independent with each other. So it's not like some scientific computing problem. The problem size is large enough to distribute them on different compute nodes. So we could map like millions of reads to one compute node, millions of reads to another compute node, and we do need the communication between these compute nodes. So they're like totally independent problem. So in this way, we could focus on the design for a single node and focus less on their the communication network. And that is one difference. And the second difference is the operation itself. For example, if we look at the NVIDIA DPX instruction. So this instruction are specialized designed for I think it's designed for genomics algorithm because in their blogs or document they use the sequence alignment of Smith-Waterman to evaluate this DPX instruction. So similar to DPX instruction, NVIDIA GPU previously does not have such support for the specialized operator used in genomic sequencing. It means the previous optimized hardware platform, like for database, does not efficiently support the operations utilized in genome sequencing. So that's why in a lot of accelerators, people has custom data paths, custom operators, ALUs to efficiently perform these operations. So these are the two main difference, I think, between the genome sequencing algorithms and the previous, like the mainstream database like applications.
Okay, yeah, interesting. But because there's too many ways to improve these speed up, like because I thought that this genome sequence search has already been solved the problem, but looks like I guess using this hardware GPU architecture you can try a different way to make it work from a single machine, looks like, right? Or or not necessarily rely on the distributed cluster, and that probably is one of the motivation. Yeah. Okay. Thank you.
Yeah, that's a good question, and yeah, it's actually open question to discussion.
Okay, so maybe I'll have one last question. What do you think? How do you envision this kind of accelerator be used in the future? For example, do you think it's part of the edge system, or you think it should belong to the cloud? For example, I mean, in terms of the DNA sequencing itself, you mentioned the precision healthcare. So in those cases, you can, I mean, if everything fits in the little box, then you can just use that thing to monitor your, I mean, personal health condition like that. That's the edge case. So what do you vision the future of this kind of accelerators?
Yeah, I think on both edge and cloud side, there are lots of opportunity. So firstly, on edge side, there are some passage years ago, there are some research work integrating some computing logics to the sequencer. And the target is not to acquire the entire human DNA from the edge device, but the only target is to filter out some parts of the sequence to detect the pathogen. For example, the COVID virus. So reducing the workload and only focusing on the important task is an opportunity on the edge device. Another possibility still on the edge device, but here the edge is not like as small as a smartphone, but an FPGA card integrated in the sequencing machine. That is a current solution like Illumina sequencer provides currently. So when they sell a sequencer, if this sequencing machine is like similar to a desktop, and they could integrate an FPGA card to accelerate the local computations. And the cloud may, I think it could be a target scenario for some, downstream analysis. For example, when we acquire the sequencing, the sequencing result, there could be some downstream analysis to analyze like for different genes or like different disease diagnose and so on. And these platforms may not focus on the sequencing itself. So for these applications, or some clinical applications that could be put to the cloud, that's another opportunity. And for some clinical applications where for example, the Illumina sequencer, that large sequencing machine with FPGA cards, are not convenient to access. Like some patients may not have opportunity to get their sample sequenced in a short time in a large hospital. So if they have an edge sequencer and they want to get the sequencing result as soon as possible, like in like 10 hours at home. So this data could also be uploaded to the cloud and analyzed. Yeah, but then in this case, probably security and privacy could be another issue. Yeah, that's mostly my vision for the cloud and edge application opportunities.
Okay, cool, thank you. So, any more questions? If no, let's thank you for the talk. And thank you for sharing your research with us. And yeah, maybe we will meet soon in Aspus. Otherwise, I think that's the end of today's talk. I'll share with I have already recorded the video and I'll upload it to YouTube and ask for your approval. Once approved, everybody can see that. Okay. Thank you again, and that's the end of today. So see you next time.
Thank you, everyone.